Hi everyone, welcome back to the Biochem YouTube channel. Today we will continue our discussion about the lipid bilayer. In the last video, we learned about the lipids forming the membrane. Now we will see how they affect the membrane as things do not simply diffuse through them. Lipids are insoluble in water, so they tend to group together spontaneously, which means this happens favorably. For example, individual units with a single fatty acid form micelles. On the other hand, bilayers are lipids aggregates in which two lipids monolayers form a two-dimensional sheet. Lastly, vesicles form spontaneously when a bilayer sheet falls back on itself to form a halo sphere. As a fun fact, vesicles are used to engulf medicines and deliver them to the inside of cells as they can diffuse through the cell membrane. Now, let's talk about membrane fluidity. There are three main factors we will take into account, and these are temperature, cholesterol, and saturated versus unsaturated fatty acids. As the temperature decreases, the fluidity of the membrane generally decreases as the fatty acids remain stable, and vice versa. Cholesterol influences the fluidity of the membrane in a bidirectional manner. At high temperatures, it decreases fluidity, and at low temperatures, it increases fluidity. It acts like a buffer since the goal is to keep the fluidity constant. Lastly, saturated fatty acids create a less fluid membrane because they are very stable structurally. Therefore, they need higher melting temperatures to increase the fluidity of the membrane, whereas unsaturated fatty acids have lower melting points since they are already fluid due to the structure. Now, the bilayer is not only composed of lipids, it also has proteins and enzymes. The different type of proteins are peripheral membrane proteins, which are associated with the membrane through electrostatic interactions and hydrogen bonding. These can be released by mild detergents or by interfering with this bonding or interactions. Integral membrane proteins are firmly embedded with the lipid bilayer, and this can be monotopic, which have small hydrophobic domains that interact with only a portion of the membrane, bitopic, which expand the bilayer once, extending on either surface to have a single hydrophobic sequence somewhere in the molecule, polytopic, which cross the membranes several times with multiple hydrophobic sequences of 20 residues, and removable only by hydrophobic agents. Lastly, amphitropic proteins associated reversibly with membranes are found in both membranes and in the cytosol. And just like that, we have reached the end of this video. If you have any questions, do not hesitate to reach out to your SI leader or go to office hours. See you soon!